Ever wondered how monoclonal antibodies are made and named? These biochemical superstars play a critical role in modern medicine, acting as our body's own personal security system. Monoclonal antibodies are synthetically manufactured to mimic human antibodies in the immune system. Think of them as tailored bodyguards, specifically designed to identify and neutralize foreign intruders such as bacteria, viruses, or even cancer cells. But how are these powerful defenders created? They're made using cutting-edge biotechnology, carefully engineered to mimic the natural antibodies our bodies produce. And here's where it gets interesting. There are 4 different types of monoclonal antibodies, each named based on what they're made of. This helps scientists and doctors quickly identify their origin and potential uses. From murine to human, each type of monoclonal antibody has its unique features, applications, and strengths. Understanding these differences is key to harnessing their potential in medical treatments and therapies. So, ready to delve into the fascinating world of monoclonal antibodies? Let's dive deeper into the 4 types of monoclonal antibodies. First up, we have murine monoclonal antibodies. These were the pioneers in the field of monoclonal antibodies, produced in labs using hybridoma technology back in the mid-70s. The name murine might ring a bell for some of you, as it pertains to rodents, specifically mice and rats, from the Muridae family, which are the hosts of origin for these antibodies. Now, if we take a step back and look at the bigger picture, we can appreciate that these murine monoclonal antibodies have played a pivotal role in the evolution of modern antibody production techniques. Their potential applications have been recognized in both therapeutic and analytical fields. In therapeutic applications, these antibodies serve as a blueprint for the development of antibody engineering techniques. This includes chimerization, humanization, and crafting bispecific antibodies from antibody fragments, known as single-chain antibody fragments, or scFvs for short. In a more specific example, murine monoclonal antibodies have been put through preclinical trials for their potential use in treating Cryptococcus neoformans, a type of fungus. These trials involve using the antibodies on the capsular polysaccharide to boost the therapeutic activity of a drug called amphotericin B. Now you might be wondering how to identify treatments involving these antibodies. Well, it's quite simple. The names of treatments involving murine monoclonal antibodies end with the suffix OMAB. Next, let's look at the chimeric monoclonal antibodies. Now, moving on to chimeric monoclonal antibodies. Imagine a blend, a fusion, if you will, of mouse and human parts. That's exactly what chimeric monoclonal antibodies are. These are structural chimeras made by merging the variable regions from one species, like mice, with the constant regions from another species like humans. But why this architectural fusion? The aim is twofold. First, to reduce immunogenicity, the ability to provoke an immune response. Second, to increase the serum half-life of these antibodies when they are prepared for therapeutic purposes. Let's delve a little deeper. Chimeric monoclonal antibodies retain the original antibodies' antigen specificity and affinity. This means they can precisely identify and bind to the antigens, much like their original counterparts. This precision and retention of affinity is a crucial aspect when it comes to their therapeutic applications. You might wonder, how do we name these chimeric wonders? Well, they have a unique identifier. The names of treatments involving chimeric monoclonal antibodies end with GMAB. This suffix serves as a telltale sign, helping us distinguish them from other types of monoclonal antibodies. In summary, chimeric monoclonal antibodies, with their unique blend of mouse and human elements, offer a promising approach in the field of therapeutic applications. Their reduced immunogenicity and increased serum half-life make them a valuable tool in the fight against various diseases. Let's proceed to the 3rd type, the humanized monoclonal antibodies. Scene script. Next in line, we have humanized monoclonal antibodies. Let's delve into the world of these fascinating synthetic antibodies. Humanized monoclonal antibodies are a sophisticated extension of the chimeric monoclonal antibodies. The process of creating these antibodies involves replacing almost all regions of the mouse antibody found in chimeric mAbs with human ones. The only exceptions are the complementarity determining regions, or CDRs, which are the amino acids that make direct contact with the antigen. So what does this mean? Well, it means that humanized mAbs have small portions of the mouse protein attached to the human protein. This combination allows these antibodies to mimic the human immune response even more closely than their chimeric counterparts. They retain the specificity of the mouse antibody while significantly reducing the potential for an immune response against the antibody itself. This makes them incredibly useful in therapeutic applications. The naming of these antibodies follows a particular pattern, like their predecessors. The names of treatments involving humanized monoclonal antibodies end in ZUMAB. This naming convention helps differentiate humanized mAbs from other types and provides a quick way for healthcare professionals to identify the nature of the antibody. Understanding the structure and function of humanized monoclonal antibodies is crucial for appreciating their role in modern medicine. Their unique combination of mouse and human components allows for a high degree of specificity in targeting disease, while also reducing the risk of an immune response against the antibody itself. Finally, let's explore the last type, human monoclonal antibodies. Lastly, we have human monoclonal antibodies. These are the pinnacle of antibody engineering, fully human proteins that have been manipulated using advanced molecular biological techniques. Here, we delve into the world of precision, where we modify the amino acid sequences to alter the specificity, affinity, or biological functions of the antibody. This process can even lead to acquiring sequences that are not part of the human repertoire, expanding the potential therapeutic applications of these antibodies. Imagine a specialist tailor who not only creates a custom suit, but also spins the fabric. This is the level of detail involved in the production of human monoclonal antibodies. We are essentially tailoring these antibodies to fit the immune system perfectly, enhancing their ability to target and neutralize specific pathogens or cells. Now let's talk about their naming. The treatments developed using human monoclonal antibodies end with UMAB. So whenever you come across a treatment with this suffix, you can be sure that it involves these finely tuned, fully human proteins. In the grand scheme of things, human monoclonal antibodies represent a significant leap forward in the field of immunology. They offer new avenues for treating a wide range of diseases, from cancer to autoimmune disorders and even infectious diseases. Now you have a better understanding of how monoclonal antibodies are made and named. Until next time, stay curious and keep learning.